Hey guys, Dr. McDonald. Let me get all straightened up here. We're going to start on chapter 4 right now. We'll see how far we can go. I do, again, as always, I'm going to keep it to about 30 minutes. So let's go ahead and jump right on in. We're talking about histology, which is tissues. Tissues are the next level up from cells. Cells are just groups of tissues that do basically the same thing, and they're more or less the same in structure, which that's our definitions. We have four main types of tissue in the human body. We have epithelial tissue, we have connective tissue, we have muscle tissue, and we have nerve tissue or nervous tissue, either one. I'm in the way, so let me move myself up. Oof, sorry guys. Nervous tissue is going to be the brain and the spinal cord and the peripheral nerves. We will talk about that toward the end of this semester. But basically, this is the guy that checks what's going on outside of us and inside of us, sends that information to the brain. The brain compares it to what it wants. The set points, the range, we're talking about this again, and makes its decision and then sends out instructions either through hormones or through nerves to cause things to happen, to make things go back. Muscle tissue, we're going to do this right after the bones. Muscle tissue does like this. Let me get straight, guys, so you can see. So I've got a muscle fiber, which is a tissue cell, which is a muscle cell, and it's got the ability to pull toward the middle. We'll talk more about that. We have three kinds of muscle tissue. We've got the muscle tissue that's in the heart, which is cardiac. We've got the muscle tissue that actually is under voluntary control. Those are our skeletal muscles. These are the guys that kind of get tired if you keep them working too long. And then we've got smooth muscle, and that's going to be the muscle that moves things through our GI tract, that squeezes. These guys a little bit longer lasting. And then we have epithelial tissue. Epithelial tissue, a lot of it is for covering it. The skin is an epithelial tissue. We also use that for glands that make things, that make products. Our sweat glands, our oil glands in our skin, mucus glands that produce the mucus that lines the GI tract. All of these. It's also used for absorbing. In our GI tract, starting with the small bowel, we're going to have a lot of epithelial tissue because that's specialized for absorbing things. We can also secrete, push things out. We'll talk more about that. Connective tissue, there's a lot of different kinds of connective tissue, and it's going to take us a little while to go through that. But connective tissue is going to protect us. It's going to be a support, and think of the bones. It's going to hold our bones together and hold our muscles to our bones. Hold on, guys. Dense, regular connective tissue. Fat, adipose tissue is connective tissue. Blood is a connective tissue. So there's a lot, cartilage, bone, all of these, and we'll talk more about those in the upcoming slides, guys. We don't get bogged down. This is just introductory. Okay, first thing we'll talk about epithelial. Muscle tissue, you cover that later in this semester. We'll cover the bones later and the nervous tissue, but this is the only time we really get in and talk about epithelial tissue and connective tissue. So we got a little bit of an emphasis on that. So we have the covering and lining epithelium. That's both on the external, the skin, and the internal, the lining of our GI tract. The lining of your mouth is a type of epithelial tissue. We also have the glandular we talked about that produce products and put them out. We'll talk more about that. The epithelial tissue have certain characteristics, and we're going to be talking about this in the lab as well. But these cells have polarity. What in the world are you talking about? All I'm saying is that when you have an epithelial cell, let me see if I can make one. One end of that cell is attached to something, and of course the sides are more or less attached. But this is the thing, you've got one end, it's called the apical end, that is facing empty space. Think about your skin, the top layer of your skin, there's one part of that surface that just faces outside. In the GI tract, there's one side that faces the hole or the lumen of the GI tract. The side that's attached to connective tissue is called the basal. So this is one thing. Epithelial tissue is all about cells. Now some of the other tissues, the cells are kind of scattered, but epithelial, they're pretty much well packed in there. They form sheets, if that makes it a little bit easier. Some of those surfaces of the apical can have those microvilli. We talked about that in chapter 3. Again, it's something for absorption. There's certain places that we have that, and every place that we have that is going to be an AMP2 topic. We'll talk about the GI tract, we'll talk about the kidneys, but that's not something we're going to be really talking a lot about on this semester. The basal lamina is, let's just leave it and say it's connective tissue. We'll talk more about that when we get to the connective tissue. Okay, here's another good way of seeing it. So there's the epithelial tissue, and you can see that one side of this just opens up into space, empty space. That's the apical, the basal, and is this side, and it's the one that's attached to the connective tissue. Connective tissue holds it on. That's basically what its function is. Okay, let's see if we got the polarity. Basal lamina. The basal lamina, well, we'll talk about that characteristic when we get to it. They have specialized ways of fitting the cells together. How do the cells stay together? Well, there's actually 3 kinds of junctions or ways of holding cells together, depending on what you want to do with them. We have something called tight junctions. That's if you want to make the space between the cells non-permeable. You want to keep things in. And there's places that we need to do that, like in the kidney again. We have the desmosomes, and that is something that we use when we want strength, when we want those tissues to be able to hold together. And then we have one thing called a gap junction, and it's really a channel or a tunnel between 2 cells so that things inside the cell, like ions, can pass from one to the other with no impediment. Oh, I'm sorry. I think I went back. Okay. No, I didn't. This is the same picture, different slide. Connective tissue has, or epithelial tissue has connective tissue under it to attach it. Call this the reticular lamina. The basal lamina and reticular lamina, usually I just refer to it as the basement membrane. And that's it. It's kind of easy to think. If you think of these guys as being like a structure, what do you have at the bottom of a house? You have a basement. You know, whether it's a crawl space or a full basement, you have a basement. So it's really kind of a good way to think about it, help you remember it. Okay, so more characteristics of these guys. Closely packed cells. That's what epithelial, that's one of their main characteristics. These guys are going to be held together either by tight junctions, if they need to be, so we don't have anything leaking between the cells, or desmosomes to hold them together, at least in the lateral direction. They all have the basement membrane. Now here's some other things. They are avascular, but they got nerves. So your epithelial tissue has innervation so we can feel things like temperature and pain and pressure, but they don't have a blood supply. Where do they get their oxygen and their glucose to stay alive? It actually diffuses up from the connective tissue. The blood vessels are in the connective tissue, and that's where this is going to be coming from. Here's another thing. Epithelial tissue, especially on your skin, we have to turn over our skin very rapidly because we're always bumping. I mean, we're shedding skin all the time. So about every 30 days, we run through the entire outer layer of the skin. So these guys have a high rate of regeneration. In other words, they do a lot of cell division because a lot of these cells are kind of made to be used. And the best way to think about that, I think, is to think of it as like brake pads on a car. You get the brake pads, you got this big, thick thing, but every time you stop, you rub a little bit off, and that's why we have to get our brakes fixed every once in a while, replaced. So these guys are kind of like that. We know that they're going to be worn off, and we have to return and do a high rate of replacement. Now, here's something else, guys. The more you divide, the more likely that the odds are that you'll get a DNA damage at some point, carcinogen exposure or even not, just randomly. A lot of the cancers that we deal with are from epithelial cells. We'll talk more about that. Okay, how do we classify these epithelial cells? Well, it's really kind of easy. First thing, how many layers are there? How many cell layers? If I only have one cell layer, it's simple epithelium. If it has more than one, if it has 2 or 1,000,002, it's stratified. So that can't get any more simpler. The next thing we classify them by is the shape of the cell. Is it flattened like a narrow rectangle? It's squamous. Is it kind of cuboidal like a square? It's cuboidal. Is it like a long rectangle? It's a columnar. And then we add the stratified or the simple to that, depending. And I think I got some good pictures. So there's a simple epithelium because there's only one cell layer. But if I look at that, I see that the cell looks kind of squashed. So that is going to be simple squamous epithelium. I look over here. Those look cuboidal, but those look squamous. How do I name this? Well, let's figure out. We got more than one layer, so it's stratified. The top layer is squamous. Stratified squamous epithelium. Here's a rule of thumb, guys. You name the epithelial tissue by the cell type that's on top. Doesn't matter if everything underneath is cuboidal. If that top layer is squamous, it's squamous. That's pretty straightforward and easy to remember, I think. So there's our squamous cell, kind of flat. This is our cuboidal, more square. This one is an elongated rectangle. Now, this isn't lab, but if I were looking at this under a microscope, a lot of times you can't see the cell boundary. Sometimes you can, and that's really nice, but sometimes you can't. But you can always see the nucleus. If you got a flat pancake nucleus, then that's squamous. If you got one that looks round, it's cuboidal. But look at this guy. He's an elongated oval columnar. That was free. What we use simple epithelium? Mostly for bringing things in, for moving things out, for filtering. Think about it. The thicker I'm sorry, I'm trying to point to the camera. Don't want to have my hands off like this. The thicker it is, the longer it's going to take to move through it. So you have this, I'm sorry guys, and you have this. Which one is going to be faster to move things through? Well, the squamous, of course, simple squamous. Although simple cuboidal also is relatively rapid. So that's what we're using these guys for, to move things in and out. Although, to be honest with you, in our GI tract we use columnar, but where do we use these? In our lungs. Those air sacs in our lungs are made up of simple squamous epithelium, so that oxygen can move out of the lungs and into the blood vessel, and CO2 can move into the lungs from the blood vessel. Oh, excuse me. Simple epithelium, they're not intended to be protective. You don't want to wear these guys out because you've only got one layer. It's not like we got other layers to take up the slack. Okay, simple squamous. Here's a photo, and I don't know if you can see. There's the capillaries. These are the air sacs. You'd have to really look at that a lot closer up, but we were going to do this in the lab. But you can see that there are those squashed nuclei there. Simple squamous epithelium. Single layer, disc shape. I don't really see anything in here that I haven't already told you. So let's continue. Now here's simple cuboidal. Now right there you can see the cell boundaries, and that's great, but you're not always going to be able to. But when I look at these guys, I'm looking at a nice round nuclei, so I know that that's got to be cuboidal, and I only see one layer of cells, so it's simple cuboidal. These guys also are involved in secretion, absorption, kidneys, some of the glands, and on the ovary. Simple columnar, again, you look here, some of these you can see the boundaries between the cells, but you can see those elongated nuclei really well. So that's pretty straightforward. Some of them may have cilia, and there's also a lot of goblet cells. Goblet cells are they're shaped like water goblets. Okay, that's why they call them that. But those are mucus cells. They produce mucus and secrete it onto the apical surface, and we do that in our airway. We do that in our GI tract. These guys also do some absorption and secretion of mucus. Enzymes, we'll talk more about the ciliated type. These are what lines the GI tract and other places. There's a lot. Now here's the kind with the cilia. I don't know if you guys can see. Okay, you can see the nuclei. Now some of these look smaller, but they still look elongated. Now, this is an exception to the rule. If you were to look at that, you would probably say, oh, the stratified columnar epithelium. No, it looks stratified, but it's not. That's why we call it pseudo-stratified. Well, why isn't it stratified? Well, not every cell reaches the apical surface, but every cell touches the basement membrane. Believe it or not, there's only one level of cells there. So it's really more of a, it looks stratified, but it's really simple. So we call that pseudo-stratified. Most of the time where you're going to see that is in the airways. And the key that's going to help you with that is not only you see it looks stratified, but you see the cilia. And for another thing, there's only very few places where you have stratified cuboidal, and stratified columnar. Stratified squamous is all over the place, but actually stratified cuboidal and stratified columnar are fairly rare, and we'll get to that too. Okay, secretion, we got all that. Now, those little hair cells, they're not really hairs, they're just little objects. These guys move in the same direction, and what they do is they push the mucus up to keep the mucus traps bacteria and dust, and they bring it up and put it to the very top of the trachea, and then we do that, and it causes that to go from the trachea, the windpipe, into the esophagus, leading to the stomach. All right, there's other places we have this. We have some in the fallopian tubes of the uterus. Again, that's AMP2, so we won't go to it. Stratified epithelial regenerate. In stratified epithelium, only the very bottom row is undergoing cell division. Everything else is not. Their main function is to protect. That's why we have multiple layers. Stratified squamous epithelium. Again, you look right there. Now that one's was just torn off. That's the way the preparation. But if I look right there, I can see a squamous nuclei. It doesn't matter that these down here look cuboidal. What do I know? I know there's more than one cell layer, and it's squamous, so it's stratified squamous. Our skin is stratified squamous. The mucosa, which is like the lining of the mouth all the way down through the GI tract, is also stratified squamous epithelium. The skin is has a little bit more hardness to it because they have something called keratin. Keratin is a protein that's like your fingernails and your hair are made of keratin. But these guys are in the cells, and we'll talk more about this when we get to the skin. Right now, we just want to know that the non-keratinized type, the mouth, the esophagus, vagina, all of these mucosal membranes are going to have those. And then, of course, it changes to simple columnar in the GI tract. Here's another exception. This guy is called transitional epithelium. Is he stratified? Yeah, but we really don't use that name with this guy. It looks like stratified squamous and stratified cuboidal. The way you identify these guys is these guys kind of make these kind of dome shapes. And some of these guys will have two nuclei. This is what happens. These guys are kind of dome shaped, and then they stretch, and then they kind of flatten out. Where do we have this at? Your bladder. As the bladder fills up, it gets bigger. I mean, it's kind of flattened if you have no urine in it, but as it fills up, it's going to get bigger. And these are the guys that are going to permit that stretching. The ureters, the urinary bladder, parts of the urethra. The bladder is good enough. Here's our stratified cuboidal. If you look, you can see two layers. I know some of these look squamous. Some of them will, guys. It depends on how you look at it. But if you look at others, that one looks kind of columnar. But if I look at all of these, the majority of them look circular. So I can say those are cuboidal cells. There's more than one layer. Stratified. The only place you got this in the body is the sweat glands and in the breast, which is a modified sweat gland. Okay, guys, stratified columnar, very rare. And again, how do I know this isn't pseudostratified? Well, I don't see any cilia up there. The pharynx, the male urethra where the urine goes out, some ducts, in some transitional areas, but basically it's pretty rare. You hardly ever see it. Glandular epithelium. So we said epithelium could make these sheets, or they could also be a gland that produces things. Now we've got two types of glands. We've got an endocrine gland or an exocrine gland. We can also have one that's the gland is just one cell, or one that has more than one cell. So let's talk about endocrine and exocrine. Endocrine just means that this guy makes whatever he does exocytosis and just puts it out into the interstitial fluid around the cell, and it eventually makes its way to the blood vessels and goes to where it's going. Now that's endocrine. Those are also called ductless. Has no ducts, D-U-C-T. In other words, it doesn't have a channel that the product goes through. Now the exocrine glands all have a tube leading from the cell, and the product goes through the tube to where it's going. Unicellular, multicellular is pretty straightforward. Ductless, exocytosis, they're transported by the blood. Exocrine, their products need to be in a certain place, not just anywhere in the body. Excuse me. Oh, excuse me. Oh. Oh, man. Must getting allergic to something, guys. Feeling sneezes. And of course, we've already talked about the exocrine body surfaces, the sweat glands, or into body cavities, or into places. All the enzymes that the pancreas makes to help us digest our food are exocrine. We'll talk about that in AMP2 as well, so it's not here. Unicellular exocrine glands, mucus cells, goblet cells. These guys both make mucus. They produce mucin, which when it dissolves in water forms mucus. It's kind of slimy. It's lubricating, but it also acts as a trap in the airways for the dust and the bacteria, as we talked about. In the GI tract, it's more there to lubricate. If you've ever had a really, really dry mouth and you swallowed something and you felt it burn as you go down, it's a friction burn. So the mucus that we have lubricates it so that it doesn't rub up against the tube and cause that uncomfortable feeling. So that's what it's for in the GI tract. There's the goblet cells that we've talked about. Microvilli, these are for secreting. So there's this rough ER, Golgi, and here's all the mucin. It's going to secrete those out those microvilli, and it's going to coat whatever tube, airway, or GI tract. Okay, exocrine glands, multicellular. These guys have got a duct, a tube. They have a secretory unit, the acinus, and those are the cells that make the product. And of course, like all the other epithelium, it has connective tissue that supports it, where the blood vessels are and where the nerve fibers are coming from. Simple glands and compound glands. I wish there was an easy way to do this, but there's not. I'll do my best, guys, but you're going to have to understand that it's going to take a little bit of time with this one. So let's just jump right on in. Oops, sorry. Let's jump right here. This is where they are. There's the unicellular, like the goblet cells. Now here's the multicellular. Now, if you look right there, you see this thing that looks kind of like a test tube. Simple, straight, tubular. You see it's got more than one cell, but it just kind of comes down like that. A simple branch tubular. So this one stalk comes off and it branches into two. Simple coiled tubular, it's basically the same as this, only instead of just going straight, it kind of twists around. Now here's the other one, simple acinar. Look at that. You notice that it looks kind of like, you know, trying to think, a vase or vase. So this one looks like a test tube, tubular. This one is simple acinar, and it's simple. Now, this is going to get a little bit more involved, so let's just kind of do it. So if I were to look at this, I would see that these are acinar. I would see that these basically all come off the same stalk or connection to the surface. So I would call that a simple branched acinar. Now here is these guys, if you look at them, they're tubular. Now if you notice, this is why we call it compound. These guys branch. I got 3 branches, and then the branches branch. That's why it's compound instead of simple. There's the same thing, but with the acinar glands. You see that these guys, 3 branches, and each of those 3 branches branch 3 times. Now this one is, that picture I really don't care for, but this one is a combination of tubular and acinar, and is compound. Yeah, it should be like simple, but they don't use that terminology. So these guys are kind of tubular, but on the very ends they turn acinar. So it's kind of a combination of the two. That's kind of a problem. Now this is going to be something else. These guys have sound-alike names. I wish they had given them different names, but they didn't. Okay. We're going to talk about 3 types. We're going to how we classify these glands by how they secrete. Merocrine is going to be, I know we already talked about endocrine and exocrine. That has to do with whether it's got a duct or not. This has to do with how it actually gets its product out. Most of them are just going to do exocytosis. You know, they're just going to put it out. A holocrine gland, like the oil glands in our skin, basically the cell makes all this product and it just keeps making it until the cell pops. The cell ruptures and it goes out. Apocrine, these guys are going to accumulate products, but only very end of that ruptures. Controversy if it exists in humans. To be honest with you, that's not the way I understood apocrine. The apocrine that I'm familiar with has to do with sweat glands, and we don't do that till we get to the skin, which is our next chapter. I know we're at about a 30-minute mark, guys. What I think I'd like to do is try to get about 35 or 36 slides. In other words, about halfway, so I don't, you know, have to make 2 or 3 or 4 of these. So there's our merocrine products. It's just exocytosis. There's our holocrine, the oil glands, and you see them popping. We've already talked about that. That's a good illustration, by the way. No, I think I will wait, because this is connective tissue, and I don't want to start a new thing. I told you a lot, and especially the epithelial tissue can be confusing. The connective tissue is a lot more confusing. So that's why I don't want to add it on to the end of this. So look, guys, I'll see you in the next chapter 4 video, and I'm going to try to complete this. Once we get past some of the connective tissue, it does get a little bit easier. Okay, guys, well, I appreciate it, and I'll see you in the next